The mitochondrion. As we've learned in introductory biology, the mitochondrion, plural mitochondria, is the powerhouse of the cell. The mitochondria's whole job is to make ATP. Remember that ATP is the main energy currency that the cell uses. Breaking ATP, adenosine triphosphate, into a molecule of adenosine diphosphate, ADP, and a loose phosphate, releases energy. This is a hydrolytic event. You use a water molecule to break them in two. And in the process of breaking them, you release energy. Whatever that ATP molecule is next to when it releases that energy, that thing that's next to it can use that energy to do its own work. There's lots of mitochondria per cell. How many depends on what the cell needs. Cells like your muscles, for example, that are constantly doing huge amounts of work, not just when you're moving, but when you're still to maintain their own muscle tone. There's constantly these little micro twitches. And to remain, to support your posture as well. Your muscles need a lot of mitochondria. This consumes a lot of energy. Other cells that perhaps don't do things with, don't have as much motion to them, may not have as much and as many mitochondria. Mitochondria are non-stationary. They can move themselves around inside the cell based on what the cell needs. I talked about this before. They used to be their own cells. They used to be alpha proteobacteria. That's a, I think that's a family of viruses, maybe a phylum. Anyway, I drive this home because, and I repeat this so much, that it used to be its own thing way back in evolutionary time, because it retains some traits of that original thing that's important to how we consider the mitochondrion behaving in a cell. Mitochondria have their own DNA. You can see these little green rings are its DNA. It has multiple copies of its own DNA. It's kept in a circular state. And this isn't just terribly dissimilar from a regular bacterial genome. They make their own RNA. Therefore, they also have their own ribosomes. So their DNA, mitochondrial DNA, can be transcribed into RNA within the mitochondrion, and that RNA can be translated into the proteins the mitochondrion needs. The mitochondrion does not need the host cell to do these tasks for it. They can also reproduce independently of the cell. They'll be provoked into reproducing during cell division, so there's enough of them to go around for the new child cells. But they can also divide on their own during the normal lifespan of a cell if the cell seems to need more energy than it has. Again, they also move. The last point was that they're non-stationary. These all help point to the fact that these were independent organisms once upon a time. If you remember much from introductory biology about prokaryotes and how they work and how they behave, it's very similar to how mitochondria behave, only they've lost some of the functions that the host cell can do for it. Like bacteria, the mitochondrion is enclosed by two membranes of its own. It has an outer membrane, which is smooth and has sort of an overall bean shape to it. That's this orange stuff. And an inner membrane, which is folded into these deep invaginations called cristae or cristae. This yellow line represents that inner membrane. And you can see how it has this wavy sort of arrangement. Now remember, why do we see folding and waviness of membranes? To increase surface area. Why do we care about increasing surface area? Because the stuff that happens in the cell almost always requires a membrane across which something can happen. The more surface area you can pack into over a given space, the more stuff you can make happen. In this case, this inner membrane is going to be really important, a really important barrier in keeping some things on the inside and some things on the outside to create a gradient that it can use later on by diffusion, allowing selective diffusion. One more thing I want to point out with having its own DNA, this DNA comes exclusively from your mom. An ovum that a woman releases is a full, normal sort of looking cell with its own nucleus, its own organelles, its own mitochondria. A sperm needs to swim, right? It needs to travel. It needs to therefore be able to burn a lot of energy. It needs to carry a lot of its own food source, and it needs to carry its own genetic material. It doesn't necessarily need to carry anything else. Its whole job is to deliver genetic material. So it loses, in the process of maturation, it loses a lot of organelles, including mitochondria. When the sperm penetrates the ovum, and then the ovum divides into smaller cells, as it develops into a fetus, the mitochondria that was in the original ovum divide along with it. Therefore, all of the mitochondria that you have in your body is maternally derived. If you're a woman, your mitochondria in every single cell in your body came from your mom, which came from her mom, which came from her mom. If you're a male, all of the mitochondria in your body, I'm speaking genetically here, all the mitochondria in your body came from your mom and her mom and her mom. There's no equal contribution of mitochondria from men and women. Now, big fat asterisk, because this is science and we're always learning something new. Very, very, very recently, as in the last few months, scientists have discovered some instances of paternal, male-derived mitochondria existing in people's genomes. This does not seem to be common, but we're still learning about it. So this maternal lineage thing I just talked about might be different by the time you get into the later scientific careers. Inside the inner membrane is a matrix. Whenever you see matrix, you're going to think of some sort of a goo. Sometimes it'll have fibers embedded in it, but it's always goo-based, thick liquid-based. So the inner membrane is filled with gel-like substance that makes up the whole interior of the mitochondrion. The aqueous environment inside the inner membrane is a little different from the aqueous environment outside the membrane. So the mitochondrion's whole job for our purposes is to make ATP, that energy currency of the cell. And it's packed with features that can help enable that to happen as efficiently and as much as possible. But first, we have to bring in stuff to feed the process of metabolism, right? We have to bring in substances to make ATP. What we do is we eat, right? We eat food. The process of digestion breaks down food into simpler molecules. The one we care about most is glucose. All other things being equal, your body prefers glucose as its energy source. So it's going to try to convert as much as it can into glucose. This glucose is absorbed into the bloodstream. This process of glucose absorption, or any simple sugar absorption, actually happens inside your mouth. It happens before you even swallow. You have enzymes in your saliva called amylase, and amylase's whole job is to break down sugars and help with their absorption through the mucous membranes on the inside of your mouth. So you start getting sugar right there. That's why if you suck on a hard candy, you're getting those calories, you're getting that sugar, but you didn't ever swallow it. It was absorbed directly through your mouth. As we discussed before, cells let glucose in primarily with the help from insulin. You also have sodium glucose pumps. That brings in a little bit. Most of it needs to be let in with insulin. You have receptors on your cells that are specific to insulin. When insulin is let in through that receptor, it drags the glucose along with it. Glucose undergoes a process called glycolysis. It's also written right here. Glyco refers to glucose. And lysis, what does that refer to? Remember, like hydrolysis, what does lysis refer to? Breaking. So you need to break down glucose, okay? The process of glycolysis is the process of breaking down glucose. This happens in the cytoplasm of your cell. We're not at the mitochondrion yet. We're still just floating around in the regular old cell. The process of glycolysis produces smaller subunits and a little bit of ATP. Some of those subunits can then go forward into the mitochondrion, get turned into a little more ATP, and ultimately powers the electron transport chain in the mitochondrion, which makes a lot of ATP. The preview to the next several slides is basically this. Glycolysis makes a little ATP. You also spend a little ATP in the process getting it started. The next step, the citric acid cycle, makes a little bit of ATP. Not terribly much, but some. The final process of electron transport chain, this is the only step that really requires oxygen to be present. This step makes a ton of ATP. Overwhelmingly, this is the process that makes the ATP in your body. So you can make ATP without oxygen, but you can only really get the full bang for your glucose buck if you have oxygen present to do the electron transport stuff. Going through this whole process of glycolysis, the citric acid cycle, and the electron transport chain, otherwise known as oxidative phosphorylation, involves a long series of redox reactions. You're going to be breaking down and rebuilding some covalent bonds. This is going to be a heavily enzyme-driven process. And as we go through this process, we talk a lot about enzymes doing things and making things happen. But remember, enzymes don't really make things happen. That's just a convenient way to say it. Enzymes simply lower the activation energy so that a chemical reaction that would ordinarily happen, it would just take a long time, can happen within a reasonable amount of time and with a reasonable amount of energy. Usually how enzymes do that is by changing the shape of whatever they interact with. They grab onto a molecule, they bend it, tweak it, rotate it, whatever, so that two things that want to interact are closer to each other. And then either breaking or bonding can happen based on how it rearranges that molecule. So the first step, I already mentioned, is glycolysis. This is a little non-intuitively laid out. It's laid out right to left. I know we usually read everything left to right, but here we are. To start glycolysis, you need to invest 2 ATP. This isn't shown. So you had one 6-carbon sugar, you spend 2 ATP, and you break it down into a smaller byproduct. This new phosphorylated glucose splits in half. This is the actual splitting part. And through a series of enzymatic reactions, all of these ACEs are different enzymes. Anything that ends in ACE is an enzyme. You're going to go through a series of oxidation and reduction, a series of redox reactions to convert each of these now 3-carbon sugars, used to be 6, glucose is a 6-carbon sugar, now we have 2 copies of 3-carbon sugars, to turn each of these into a molecule of pyruvate. So one glucose gets split into 2 glyceraldehyde 3-phosphates. Glyceraldehyde 3-phosphates, each of which turn into a pyruvate. One glucose into 2 smaller 3-carbon sugars. Each 3-carbon sugar gets converted to a different 3-carbon sugar called pyruvate. The end result of this process is that you have reduced NAD, which is an electron carrier, into NADH. This is going to go forth into the electron transport chain and be used there. You create 2 molecules of ATP per conversion from glyceraldehyde 3-phosphate to pyruvate. So in the beginning, you spend 2 ATPs, 1 ATP to make each of the G3Ps. And for each cycle of each G3P turning into a pyruvate, you make 2 ATPs. So you spend 2 ATPs right out front, you make 4, net gain of ATPs is 4. You also end up with byproduct of 2 NADH molecules. Again, it's going to come in handy later. A water molecule during a part of hydrolysis. Oh, I'm sorry. There's two two H2Os end up getting made, one per conversion of glyceraldehyde 3-phosphate to pyruvate. Okay? So in this very first step, no oxygen is needed. You make a little ATP, not much, but some. So, I mean, it can be used by your cells to make some energy. You make a little ATP, you make a little bit of a product that can be used later on to make more ATP, and you make a little water. The waste product of glycolysis is water. If oxygen is present, mitochondria can use these products to make even more energy. The next step after glycolysis is referred to as the citric acid cycle, also known as the Krebs cycle. I will traditionally use the term citric acid cycle. You could use either one. You may see it listed as Krebs cycle. I use citric acid typically. In this case, each of those pyruvates, remember that was one of the end products of glycolysis, and NADH enter the mitochondrion's matrix. So you're going to go in and start doing stuff along here. Pyruvate is oxidized to become acetyl-CoA. Just memorize that part. Pyruvate gets oxidized to become acetyl-CoA. NADH picks up electrons and loses a carbon. In the process of losing a carbon, it's going to bind to oxygen around and become CO2. One of the byproducts of metabolism in your body is carbon dioxide. You exhale carbon dioxide. This is where that carbon dioxide comes from. Each pyruvate will then move through the cycle, the citric acid cycle, to produce 4 NADHs, an FADH2, this is going to become important in the next step, an ATP, and 3 carbon dioxides. I don't need you to memorize every step. I have videos that I've posted at the end of this unit, at the end of this module, that will take you through each of the steps to make all of this a lot clearer. You really should look through those steps to refresh it. I'm going to ask you on the exam about the take-home messages. What parts make a lot of ATP? Which parts make less? Where do you need oxygen? Where do you get rid of carbon dioxide? What stages make water? How much energy is made in each case? What happens if you take oxygen away? What happens if, of other circumstances, okay? What happens if you have no mitochondria? What can you still do? I'm not terribly concerned about you knowing every step of the way, but you should know the parts that I do put on the slides. So you have pyruvate entering the citric acid cycle. The citric acid cycle needs oxygen. One thing you make is acetyl-CoA. You make carbon dioxide, that's what you're exhaling, a little bit of ATP, and molecules that can either go back and fuel this process or go forth into the electron transport chain. Mitochondria make most energy by the electron transport process, also known as oxidative phosphorylation. So here you have the process of glycolysis. Glucose came into the cell. You have glycolysis, end product, a net 2 ATPs and 2 pyruvates. It's only showing one here because it's going to just trace the path of one of those pyruvates through the mitochondrion. The pyruvate enters a mitochondrion and goes through the citric acid cycle along the inner membrane of the mitochondria. You make a little more ATP, you make some carbon dioxide, you make a little more NADH. You also make a little FADH2, which is going to have its own role in the electron transport chain. The electron transport chain also happens along the inner membrane of the mitochondrion. The fact that this material outside the inner membrane and this material in the matrix is a little different is important. It's also important to remember that the inner membrane is a lipid bilayer. Enzymes are embedded in that lipid bilayer, and things that aren't specifically allowed to go through the lipid bilayer, particularly things that are not fat soluble, are going to be, the lipid bilayer there is going to act as a very firm barrier between the inside and the outside of that inner membrane. So if you're doing something where you're pumping things out into this intermembrane space, they're going to stay out. If you're doing something to pump things into the inner membrane, they're going to stay in, unless you give them a specific designed outlet to go the other direction. Again, this is where the most energy is made. You can make energy with glycolysis, or the ATP, the energy currency. You can make energy, the energy currency, during the citric acid cycle. But this, the electron transport chain, is where you overwhelmingly make the most energy. NADH and FADH2 move to the inner membrane. We're in the matrix in here, and this up here is the intermembrane space. So here would be like the outer membrane of the mitochondrion. Okay? You have 4 transport enzymes embedded into the membrane, the inner membrane of the mitochondrion. You have 1, 2, 3, 4. You also have at the end a proton pump and a final enzyme to get the energy molecules where they need to go. What these do, these first 4 proteins, these first 4 enzymes, are going to use energy to pump hydrogen ions into the intermembrane space, creating a concentration gradient. So it's going to pump protons out. Protons, once they're on one side of this membrane, can't come back in unless it has a specific way to be allowed in, a specific door. So these guys are going to donate electrons to the enzymes, and effectively the enzymes are going to use the shuttling and bouncing around of electrons to fuel their pumps. Remember, electrons are basically energy. They're pure, they're rich in energy, and movement of electrons can fuel things. It's basically electricity. So these guys donate electrons. The electrons come in and zip, boom, 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 down this chain. You have other molecules that help shuttle them too. Okay? But at the last step, you're going to use oxygen to harvest your electrons. You want oxygens to enter the system in one place and exit at a specific other location so that it can't go through and transmit energy to things that don't want energy to be absorbed. Oxygen is reduced and accepts hydrogens. When that oxygen gets an extra electron, it's more inclined to bind, and it's going to bind to loose hydrogens, loose protons that you find in here. That's going to make H2O. An oxygen gets an extra electron. It's then going to be able to attract two hydrogens to it to be complete. Now remember, through these processes, this movement of electrons is going to fuel protons to be pumped to the outside, so that there's going to be more protons out here than there are in here. One of the other ways that we reduce the number of free protons in the matrix is by binding them. Proton's just a loose hydrogen ion, just a loose hydrogen with no electrons. One way that you're going to reduce the concentration of protons in the matrix is by binding them up to oxygen to make water. So not only are we shuttling protons out in this process, but we're also binding up hydrogens that are on the inside so that they're not free to do other stuff. This is why water is another byproduct of metabolism. You make some water in the very beginning in glycolysis, and you make a little more at the end in the electron transport chain. Now you have lots of protons, hydrogen atoms with no electrons on them, and very few in here. So at the end of this chain, where you shuttled a bunch of protons out and created this artificial gradient where you have a high concentration of protons on one side and low on the other, you put a special door through which protons can move. By basic processes of simple diffusion, by basic concepts of simple diffusion, molecules want to move so that their concentrations are equal on either side of a membrane. There's a high pressure of protons up here versus here. This pump allows protons to move through it. And it's called a pump because when protons move through it, it turns part of that molecule like a turbine. This is exactly how a dam works, only instead of protons, you have water. Hydrogen ions get shuttled through, protons get shuttled through. When it rips through that molecule, it's able to turn the turbine part of this molecule. When that turbine turns, it permits ADP and a nearby loose phosphate to bind into ATP. By this process, you've converted ADP into ATP, ATP being the primary energy currency of the cell. Finally, you want to continue fueling this process, and to make sure that ATP is usable, you want to get the ATP out and bring more ADP in. You have a final channel here that allows an ATP to be exported for every ADP that's brought in, so that you can keep refueling this end process. This makes 32 to 34 molecules of ATP for every glucose that you consume. So one glucose makes, in the beginning, 2 in glycolysis, 2 ATPs, right? 2 ATPs in citric acid cycle, because you have that pyruvate going around, you have 2 pyruvates and you go around twice. And 32 to 34 in electron transport chain. So total, you could have up to, let me pull that down so you can see, up to or equal to 38 ATPs per glucose. Okay? One glucose, because of this mitochondria, is able to generate a ton of available energy. And this is happening hundreds and thousands of times per second in every mitochondrion in your body. It's an extremely fast, efficient process. It's a really efficient way of turning the food that you eat into usable energy. Now, other things that you consume, anywhere from the closely related sucrose to big complex proteins, starches, amino acids, fats, can all be broken down and put through some variation of this process. But it requires an input of energy to do. As a broad rule, the further away molecularly something is from being a glucose, the more energy you're going to have to put in to turn it into energy. So a starch, which is just really a long string of glucoses, you need to burn a little energy to break that up, but not terribly much. That can get turned into glucose fairly readily and fuel a lot in your body, make a lot of energy. Something different, like proteins or fats, you need a fair investment of energy to turn those into simpler products that you can use to fuel digestion. And that's going to require a little bit more. Because of that, they don't necessarily have the same type of energy payoff in your body that sugar is molecule for molecule. There's a video at the end of this unit, at the end of this module, that shows all of these processes really clearly, step by step. It goes into a little more detail than you would have gone into in introductory biology. The extra detail you didn't go over in Intro to Bio isn't stuff you need to learn, like the F1, FO components of the ATP pump. You don't need to remember that. But I do need you to go through and watch those videos to get the point of glycolysis and to see redox in action, and to learn the end products of glycolysis and why they happen, to learn about the process of the citric acid cycle and see the redox reactions that are happening to turn that the sugars into different versions of the sugars, and how turning each version of the sugar into a different version of that sugar can add or subtract different hydrogens or electrons and make different byproducts. And finally, go to watch to really be able to visualize the process of the electron transport chain and how the movement of these electrons, how oxidation, how the electrons being lost from NADH and FADH2 to shuttle through here. Remember, lose electrons, oxidize, hence oxidative phosphorylation, can be used to make water inside of the cell, can be used to make a concentration gradient where there's lots of protons on the outside and few on the inside, how those can be used to make ATP based on principles of simple diffusion. Okay? I also just think that the illustrations that I found, thanks to the miracle of modern animation and YouTube, are really, really clear at showing where things are in the cell and the mitochondria as they happen. I think it's a lot better than traditional hand-drawn diagrams at illustrating the movement of these processes.